She is Natalia de Val. She will talk about an introduction to electron microscopy, a powerful technique for characterization proteins and pathogens. So thank you, Natalia.
So thank you, everyone. Perfect, thank you. So I am the last talk of the day, so hopefully you will not fall asleep. So my talk is very, very different from the talks like we are, listen to probably yesterday and today. So it's all science focus. So the thing I wanted to mostly explain to you guys, like I know the majority of you have a small experience or you are starting to have some experience with electron microscopy. So I would like to introduce you, how to proceed with this technique and the beautiful images that you can get with this technique and how much, science you can understand with it. So first of all, this is the typical landscape of imaging techniques, and you can see they are all overlapping. So again, if you really want to understand your biological sample completely, you will need to probably use all of them, okay? So for example, light microscopy, super resolution, like everyone here is super familiar with, they are very good techniques to have a huge or large field of view, but of course you are losing the resolution. Same with conventional SEM or TEM, so scanning electron microscopy and transmission electron microscopy. On the other hand, here you can see you have the other side of the spectrum where you have the NMR and the crystallography. Like they are very good techniques for having very high resolutions, but you are only seeing a very small area of interest. So the field of view is very small. So the beauty of electron microscopy, and this can be electron tomography as well as single particle cryotransmission electron microscopy, is like you can still get a very high resolution, but without losing the field of view. So it's a very good balance, in between both techniques. Again, even if I have a big bias for electron microscopy because I was a crystallographer when I did my PhD, but I transitioned to electron microscopy almost 20 years ago. So again, big bias for this technique. I love it. And I think it's very powerful, but I highly recommend it to, again, have access to every single technique if you really want to understand your biological problem. The more techniques you can use, the merrier. Okay, because everything is overlapping and everything will help you to understand what is your final goal. So structural biology techniques, and this is very interesting to see the evolution of, crystallography, NMR, cryo-EM. As I said before, I did my PhD in crystallography. I was a crystallographer, and it's still, one of the main techniques for structural biology. You can see NMR, is kind of a plateau right now, and you can see, cryo-electron microscopy in yellow is becoming very important. So we are expecting in the next 5 years, so even less than that, to increase even dramatically, even to reach the level of crystallography. Right now, if you go to the ENDB or the PDB, you can already see plenty of entries of new proteins, mostly small proteins or membrane proteins, like they are coming from, cryo-EM, single particle analysis. So again, very important technique for structural biology. Let's focus right now a bit more on cryo-EM. So cryo-EM, you have 3 main techniques. First one, single particle analysis, and I will explain more how it works. So you can get the structure of isolated proteins. So this is the technique like everyone uses if you really want to have very high resolutions, near atomic resolutions. The disadvantage with single particle analysis is like it's still isolated proteins, it's ex situ technique. If you really want to understand what is going on with the protein of interest in the context of the cell or tissue, you will need to do cryotomography. So that's very cool because you will be able to understand how that protein is interacting with other proteins in the context of the cell. So all the sociology of the protein, that's very, very important. And microelectron diffraction. I don't know who is familiar with micro-EV here. wow, okay. So I don't want to spend so much time in that technique because probably it's not your main goal, but I think it's important for you to know, like microelectron diffraction is a technique like it's evolving very quickly right now too. So it was developed by Tamir Gonen. It was in Janelia Farms first, and he moved, I think, probably 4 years ago to UCLA. And it's a mix in between crystallography and electron diffraction. So you are working with very, very small crystals that they are not well or they are not good enough to go to crystallography because they are very tiny, but you can still use them for diffraction under the transmission electron microscope. So the resolutions you can get, they are the same resolutions like with crystallography. So again, it's very cool because for crystallographers like me, like when I was working sometimes with membrane proteins, like I was like, oh my gosh, this never crystallized, or I had very small crystals and it's very frustrating and I cannot do anything with these crappy crystals, you know, don't worry. Don't put those crystals in the class because you can use them right now for micro-ED. So you put those crystals in a cryo-EM grid, you put them in the transmission electron microscope under diffraction mode, you localize the crystal of interest and you start collecting data. Very cool, okay? So my main focus for today is gonna be probably single particle tomography, but I will go at the end of my presentation a tiny touch on micro-ED only for your information, okay? You never know, maybe someday you will be able to use it. So again, I know plenty of people here are very familiar with the traditional room temperature transmission electron microscope. So I would like to go a bit through this technique. I don't want to spend so much time in this. You are probably already all very used to how to prepare the samples, cells and tissues for transmission electron microscopy and scanning electron microscopy. So it's still a very long process. When I was a core director in NCI, I was directing 2 labs. One was the classical TEM lab with all this process, and the other lab was the high resolution lab, you know, with micro-ED, tomography, single particle cryo-EM. So very different approaches. And this process is still, probably these protocols are 40 years old. I know exaggerating, and they didn't change, nothing. So it's still a long process. You need to prepare the sample, you need to prepare the samples, do the fixation, that can take 5 hours, the calcification, another 5 hours, staining, the hydration. I mean, it's a very long process. Some examples that you can get if you do that, you know, this is a transmission electron microscope. And these are thin sections of resin embed material. So you can see this is a 120 kV voltage transmission microscope, and you can see you can work with different magnifications. For example, the image here on the left side is only 940x. This is a medium magnification, and this is a high magnification. Those are the exposure times. The camera you have is a 4K by 4K, and you can see the quality of the images. So very high quality and very high detail. This is a kidney tissue. So again, it's a thin section on the same process, resin embed material. And again, 120 kV voltage microscope, low magnification, middle, and half magnification. So this can already provide you with plenty of information that you will find very useful. Brain tissue. Plenty of people are working with brain tissue. Again, 120 kV, different magnifications. You can see even a beautiful mitochondria with a crystal is very, very cool. More examples here, the liver tissue is like 100 nanometer plastic section. And again, we are working with different magnifications on 120 kV. If you are looking to increase the contrast, because when you are working with, room temperature TEM samples, for example, pathology labs, they are not looking for resolution. The thing like they are looking for is the contrast, to have a nice contrast, so they can visualize all the different organelles. So in that case, you can always reduce the voltage in the microscope. Instead of using 120 kV, you can use 80 kV. And that will give you the contrast that you will need, if you need more contrast. Another example, we are working as well with plants. In the US, we have more and more people working with plant biology. And for example, this is a tobacco plant tissue, 80 nanometers plastic section. And again, you can see the differences in between low, medium, and high magnification. So that is all the classical TEM, process. And again, depending on your research project, probably that will be enough, and probably that will be the only thing you will be doing. Transmission electron microscope, also scanning electron microscope as well. For people like that are a bit more adventurous or they need a bit more resolution, you need to go for cryo, okay? Because cryo-EM is the way to go to be able to visualize your proteins almost in a native context without any artifacts coming from all the fixation, the hydration process. So that's very, very cool. So the first thing is cryo-EM single particle analysis. Again, as I said before, you need to express and purify the protein of interest, so it's ex situ, and you need to be able to do all the process, and I will have another slide later where you will have all the pipeline for this. But advantages of using cryo-EM single particle versus other structural biology techniques, several. First of all, you can catch in the same experiment different conformations. An example of this is in this example. This is a ATP synthase. As you know, the ATP synthase are very, dynamic, and it's their role, it's an enzyme, so they are very dynamic. We work in collaboration with John Rubinstein and Nico Grigoriev, and in only one grid, only one cryo-EM experiment, we were able to collect data, analyze computationally the data, and separate all those 7 different conformations and create 7 different 3D reconstructions that you can put together like in a movie like this. So you can already visualize all the dynamics in the protein. So it's very, very cool. This is almost impossible to do by crystallography because sometimes when you have a crystal, the crystal is usually in one conformation. So it can be the closed conformation, can be the open, can be in the middle. So it's very difficult to have these dynamics. So this can be visualized by cryo-EM single particle. You can work as well with protein complexes. Again, the more complex or the more proteins you want to crystallize in the same experiment, the more heterogeneity you are adding into your system, the more complicated it's going to be to have a crystal, okay? So in this case, you can work with a protein, for example, an antigen, you can buy different antibodies, you can make a huge micromolecular complex with different proteins, A, B, C, D, and put that in the same experiment and have a 3D reconstruction of all those proteins together. So again, very, very cool. You can work as well with low affinity binders and even capture the post-translation modifications. That is on the scientific side. Let's talk about logistics. As I said before, I was a crystallographer, and for several years during my PhD, I was trying to crystallize, lucky me, membrane proteins. I don't know why I decided to do that. Of course, never happened. So you know what? The beauty of single particle is like you don't need crystals. Yes. So I was like, wow, you don't need crystals to do the experiments. No, nothing. And you need very small amount. Sometimes in crystallography, as you know, when I was working again with the PI in crystallography, it's like, I need 10 milligrams per milliliter of this protein. I was like, are you kidding me? I cannot even produce 1 milligram per ml. How you want me to produce 10 milligrams per ml? That's crazy. So the amount of protein you need sometimes to crystallize is very huge, and some systems are very difficult to produce. So in this case, single particle, you need very small amount. So that is very cool as well. So this is and I have the video, and hopefully this time is working so you can see the whole process. So this is the same slide like I presented a bit yesterday. So the first thing you need to do, again, is the sample preparation. That's very important, because it's the biochemistry department. Once the sample is ready, you will go with data collection in the microscope, and after the 3D reconstruction. All this part is software, is computational, so it's quite easy to do. That is very easy to do right now once you have the microscope in-house. Again, can be expensive to have the first microscope, but you can try to collaborate with people like they already have one. So it's the way to go, or with course, that's the way to go too. So the more difficult part is still, the bottleneck is still the sample prep. Okay, we are working very hard with different systems to make this more in a more accurate way, in a way like can be much more reproducible, but it's still complicated. So it's still the bottleneck of the technique. But let me show you this tiny video. I can explain to you how it works. So all those little guys moving around, those little whites are proteins, okay? Micromolecular complexes. And those proteins, where they are expressed and purified, they are vitrified at minus 180 degrees Celsius in a small cryo-EM grid, like it's a copper grid, 3 millimeters in diameter. So it's very, very small. That cryo-EM grid is going into the transmission electron microscope, and this is a typical image that you will be able to get under cryo conditions. So all those little shadows are proteins, okay? So the more images you collect, the more particles or proteins you will have. So the beauty here, and all this is done computationally with different softwares, is to have and create 2D projections, 2D classes of the protein of interest in different orientations, bottom views, side views, top views, okay, and in between. You put together all those 2D projections of your protein of interest to extract the information you want from those 2D classes to create the 3D volume of your protein at the end. And the first time you can see your protein in 3D is the best day in your life. Believe me, I was so happy when I was like, oh my God, after all these efforts, purifying the protein, going through the data processing, data collection, etc. I can see my protein in 3D. This is cool, you know, and you can play around with it. And in the lab at Scripps in San Diego, we even have these special computers where you put special glasses and you can see the 3D real coming out. It's very cool. It's very, very cool. So it's very rewarding, you know. So how to do that, okay? So I talk about the vitrification process, and that can be, again, a bit tricky. But once you have your protein purified and it's homogeneous, not falling apart, etc., you can vitrify it. And that is done with this instrument, like it's called the VitroBot. And it's a very cool instrument. You have here the area where it's a chamber where you can control with this iPad here, the temperature and the humidity. Usually proteins are happy when they are vitrified at 4 degrees Celsius in the chamber and 100% humidity, okay? But you can change those parameters because, again, sample dependent. Depending on the protein you are working with, maybe some proteins that are more happy at room temperature in the chamber. So you need to play around with those parameters. So you can change all the parameters here, and this is the iPad you will be able to see in here. This is a chamber. And if we zoom in the chamber, you will be able to see these filter papers. Those are the filters papers that you will use for blotting. So this is a tweezer, and this is the grid. So if you zoom in that area, this is your tweezer, and this is the cryo-EM grid. So it's a 3 millimeter copper grid. So it's very tiny, okay? So you apply on the side your sample of interest, usually is 15 microliters. So again, very small amount you need per grid, so it's very convenient. And usually you wait probably 5 to 10 seconds, and this is the timing here, you know, wait time. And after the waiting time, you are blotting with those filter papers. So what that means, you are, you know, approaching those filter papers into the grid to remove the excess of liquid, because you want to generate a very thin layer of buffer, like it's gonna be around your protein of interest, but not too thick, not too thin. So that's the tricky part, okay? And once you are doing that, the system automatically will plunge this into the liquid ethane. This is the cup with liquid ethane at minus 180 degrees Celsius. So that the vitrification is so fast, like you don't have any crystalline ice. It's vitrified ice. The crystalline ice is the regular ice that you don't want to have in the grid, because that's the thing that we call ice contamination, okay? So once you have that, you have the perfect sample, all the biochemistry department, you are able to vitrify the sample, very cool, you have the perfect ice, the perfect particle, etc. You go to the microscope right now, okay? So of course, when you are working under cryo conditions, every single thing you are touching that grid needs to be under liquid nitrogen, okay? So once that cryo grid is vitrified, you need to store it in a dewar with liquid nitrogen. And you can store that for months, no problem. When you are touching that grid to transfer the grid into a transmission electron microscope, every single tweezer, every single thing like is touching that grid, is gonna be under liquid nitrogen conditions, okay, to avoid any ice contamination. So you have already the grid in your microscope, and the microscope is cooled down as well, okay? And this is a typical atlas, so it's a tile series that you can take of your grid. So a grid, as we know, is a 3 millimeter copper grid, it's a circle. Inside the grid, you have squares, as you can see here, and inside every square, you will be able to see you have holes. And those holes are the ones that we are using to collect data, okay? The software like we have is called GPU. It's a software like Thermo developed, and it's very convenient because you have different colors depending on the thickness of the ice in your squares. So for example, in this case, the purple ones are thin ice. You know, the orange, medium, and the kind of blue is thick ice, okay? Generally, you want to go for the thin ice squares, because the thinner the ice, as long as you can see the particle of interest, the better contrast you will be able to get, and the better you will be able to see your particles in the transmission electron microscope, okay? So the first thing I will do, you know, when you go to the transmission electron microscope is to do the atlas, to see the quality of your ice around the grid, and to check some squares with different ice thickness. So the first one I want to go is with a square here with thin ice. This is a square. You can see inside the square you have holes. If I go to a hole right now, those are my particles, okay? Those are proteins. But what is the problem here? All the protein is stuck in this area. I have nothing here. So the particle distribution is not perfect. I will be like, okay, I can still collect data on this, but I will see if I can get better. So I can go back to the atlas, you know, the low magnification image, and I can select right now a grid or a square with a thick area, you know, with a thick ice. And if I take a picture right now, perfect, win, you know. So you can see here, the protein is evenly distributed on the ice. So every single one of those guys are different proteins. Okay, so right now I can start collecting data in this particular grid. So some examples of the data that you can obtain. And we have different instruments. Okay, we have the 100 keV voltage transmission electron microscopes, the 200 keV, the 300 keV. So the 100 keV is like a Porsche. Okay, it's already a very good car, but you can get better. For a 100 keV microscope, the resolutions here, you know, are already very high. So for example, this is a 330 kilodalton, you know, channel. It's a membrane protein. We can already get a 4.2 angstrom resolution. And this is the 3D reconstruction you can see. So it's already very, very cool. We have another, oh, sorry about that. I went too fast. We have another here, is a 200 keV, you know, membrane protein, the GABA receptor. GABA receptor is a very important target for pharmaceutical, you know, vaccines and drug development, because GABA plays an important role in plenty of different diseases. So plenty of people are focusing on this protein. So having the 3D reconstruction is super helpful to develop new drug designs. 3.4 angstroms, so not bad. The proteasome are 3 angstroms. You can see the ribosome here at 3.5. You know, the AAP6 at 3.5 as well, and the apoferritin at 2.6. So again, depending on what kind of question, biologically speaking, you want to answer, these resolutions are probably more than enough, okay? Other people is like, yeah, well, I want to do some kind of structure-based drug design. For that, you really need to have a very, very high resolution, almost atomic resolution. And for that, you probably need to go to the 200 keV microscope or the 300 keV microscope. So for example, this is the kind of results you will be able to get with 200 keV instrument. And again, if we compare with cars, this is already probably a Ferrari, okay? So much better instrument. So the same proteasome, like before we have around 3 angstroms, with the 200 keV, you can already get at 2.1 angstroms. So you increase dramatically the resolution. The apoferritin, like before with the 100 keV voltage, we have only 2.6 angstroms. Right now you can go to 1.6 in less than 9 hours of data collection. The GABA receptor 2.4, the GPCR protein, again, membrane protein, only 150 kilodalton in size, 2.6. The cyclin, this protein is very important. It's an enzyme, plays an important role in cancer. It's only 85 kilodalton. For single particle standards, it's very small. The smallest you can visualize right now for a protein in transmission electron microscope is around 50 kilodalton, 50. It's the size of an FIB. Less than that, you can try it if you are quite crazy. I will try it. Why not? You never know if it's going to work, but it's not going to be easy. I can tell you that. Okay. So 85 kilodalton, 2.5 angstroms. So it's already very good. Even smaller, 64 kilodalton, hemoglobin at 3.4 angstroms. So very, very cool results. What happens if you are working with the best instrument right now in the market, the 300 kV voltage, So you are producing the electrons very, very quickly. So that means the contrast is going to be probably better, and mostly the resolution. The resolution is going to be much better in the 300 kV versus the 200 versus the 100, okay? So if you are really pursuing the maximum resolution you can get, the 300 kV will be the best option for you guys. But again, depending on the project. And it's always heavily sample dependent, okay? Depending on your sample as well. Some samples, biologically speaking, you will have a limitation in resolution. And it's not because of the hardware, it's because of the biology of the sample. And that's the reality. So for example, the GABA receptor, you remember we are around 3 at 100 kV, around 1 point, something. With the 200, we have 1.7 at the 300 kV. And you can already start seeing small molecules, like they are drugs, like they can bind in different, areas of the GABA receptor. And you can really visualize the density of those small molecules. So this is super important for pharmaceutical companies when they want to develop new drugs, new products, and they want to understand how these products are binding into this protein, okay? And how this is affecting, the conformation of the protein. The hemoglobin, again, 2 angstroms, streptavidin. I was telling you, the limit is probably 52 kilodalton or 50 kilodalton. You can see we have a 52 kilodalton here protein at 1.7 angstrom. So it's very, very cool. And of course, SARS-CoV-2, this is a very small membrane protein, 62 kilodalton at 2.1 angstrom. So it's quite impressive. So again, single particle is the method to go, cryo-EM single particle, to reach very high resolutions, almost atomic resolutions in some cases. But the problem or the disadvantage is still ex situ, okay? You still need to express and purify your protein of interest, put that in a cryo-EM grid, go to the microscope, collect data. Some people is like right now, more and more people is like, okay, it's very cool to have the high resolution of your protein of interest, but what happened with that protein in the context of the tissue, the context of the cell? Have a question? No? Yes. Yeah, go ahead, please. It's connected. Yes. Oh, that's the 3D reconstruction map. That's the map. When you are doing the 3D map, computationally, you have always that. You can see the side chains of the protein, the beta seeds sometimes, or the alpha helices, and you will see always that this connection is the way it is always. Yeah.
Okay, thank you. And the second question is, how do you make the cryo-EM of membrane proteins? Do you use some liposomes, or do you?
Very good question. So you can have several methods. Some people express and purify the membrane protein using detergents, DDN or other detergents. Some people use amphipols. Some people use nanodisc. So depending on your protein, one method or one approach can be better than others. I know a lot of people like the nanodisc right now, because in the nanodisc, you can really change the lipid composition of the nanodisc, and you can really, adapt the best composition for your protein of interest. So I would probably recommend nanodisc if you can, but otherwise amphipols or detergents are the way to go too.
Nanodiscs are sort of rings?
Yeah, they are small rings like membrane.
Okay.
Yes. And you can test the lipid composition on those nanodiscs.
Thank you.
Of course. Yes, more questions? Yes. The magnification in the microscope? Oh yeah, so you can change that very quickly in all the transmission electron microscopes we have. It's very easy to go from low magnification to call it the atlas, to medium magnification to half magnification. The higher the magnification, the smaller the pixel size will be in your camera. It's a bottle-mounted camera, and that means like the highest resolution you will be able to get. Obviously, if you go very crazy with the magnification, you will have very small number of particles per image. So you need to have a balance where you still have enough particles per image, but the pixel size is small enough that you can get a high resolution. The limiting resolution is always, in theory, the double of the pixel size. So if you are working with a pixel size of 0.77, if you do the double of that, the limit maximum resolution that you will be able to get is 1.5. Makes sense? So you play with that. High magnification, pixel size, and the maximum resolution you will be able to get. And of course, the pixel size depends as well of the camera you are using. Of course. Okay. So cryo-tomo. This is very cool. Okay. I love it. Seriously, I love it because, again, it's visualizing the protein like you want in your cell. And I mean, that's the beauty of this, because sometimes you have surprises. Sometimes you are working with an isolated protein and you think it's a monomer, and you are convinced it's a monomer. And after you go to the cell or the tissue and it's like, oh crap, it's a dimer. And probably the dimer is the correct one because it's in the context of the cell. It's the real thing, the real stuff. So how to do that? So unfortunately, the process is a bit more challenging, okay, like single particle analysis. And I will explain to you how. So the first thing you need to do is growing the cells in the grid, okay? And that can be tricky. Depending on the cells you are working with, can be HeLa cells, can be, bacteria, whatever you want to work with. Depending on the cells you are working with, for example, neurons can be very tricky to grow on the cell, on the grid. So that can be the bottleneck part. As I said before, for single particle analysis, the bottleneck is still sample prep. Here is the same case, and it's even more complicated because you are working with cells or tissues. So let's see you are lucky and your cells are growing happily in your grid, okay? So after that, as you know, the cells are very crowded. It's like the metro in New York, rush hour. Plenty of things going on in there, okay? So if you are focusing on one protein, you really need to target and label that protein of interest in some way. Okay, you can use a fluorophore, you can do whatever you want to use, usually are fluorophores. So you target that protein of interest with a fluorophore, and you need to use the light microscopy, like you are experts or here, to visualize that. But the beauty is like right now, you can correlate that light microscopy result with electron microscopy. Because electron microscopy, as you know, we are not very fancy, it's all black and white, okay? So how to localize the area of interest is because we can correlate right now the light microscopy results with electron microscopy, and that's super cool. But first of all, once you have your data by light microscopy, you will need to do the focus ion beam, okay? How it works. As I said yesterday, the cells are very thick, If you are trying to go directly from the cell on the grid to the transmission electron microscope, you will be able to see anything. It's going to be a black box in there. Because again, the electrons cannot go through all the cell because it's too thick. So the first thing you need to do is create the lamellas. Like that has been done in another instrument, like you need to add into your pipeline, like it's called the cryo-FIP-SEN, where you are using the focus ion beam to mill the material like you don't care about, and the SEM, the scanning electron microscope to image those areas, okay? So those lamellas are very thin, usually in between 150 to 250 nanometers, And those are the ones that you will be able to transfer into a transmission electron microscope to be able to collect data and have the 3D reconstruction in situ of your protein of interest. So I will play this video here. You will be able to see. In this particular example, we are using, for the video, we are using viruses, because for viruses, if you want to work with the whole virus, you will need to do tomography, okay? So you purify the virus, you apply the virus in your electron microscopy grid, you vitrify the virus the same way you vitrify the protein the same way, you transfer the grid into a transmission electron microscope. And another big difference in between single particle and tomography is the way how you collect the data. In single particle, you don't tilt the stage in the microscope. In tomography, you are tilting the stage, usually from -60, -70 degrees to +60, +70 degrees. So you have increments, the 2 or 3 degrees in all those images. So you are creating a movie, kind of a tilt series of all the virus or all your cell of interest. So that movie is the thing that we call the tomogram. Once you have your tomogram, you need to process all the data, doing tomogram and subtomogram averaging, and we have different softwares to do this, to create at the end the 3D reconstruction. In this particular example is of the virus, but you can work as well with the lamella from the cells, okay? The process, again, in the software level, single particle analysis is much more evolved, like tomography. Tomography is still kind of a lot manual steps. Specifically with the virus, is still easy target, but if you are working with cells and lamellas and organelles, you will need to do segmentation work. And the segmentation work, again, even if we have a program called Amira to do all the segmentation work, it's still plenty of steps like they are manual, like you need to be there to do all that work. So we are trying very hard to automate and make your life easier, for tomography purposes as well, to become to the level of single particle analysis. So tomography can be done on very different samples. So as I say, you can do proteins and viruses if you want. You can do bacteria. As long as the sample is less than 300 nanometers in thickness, you can directly plunge those samples and vitrify it, the way I explained with the VitroBot, a few minutes ago. But when the sample is thicker than 300 nanometers, you don't have any choice. You need to do the cryo-feed milling, okay? So you need to do the focus ion beam to be able to mill the material and create the thin lamellas. Otherwise, you won't be able to see anything. That's the thing that happens with the cells. When you are working with tissues, the same process. Tissues are even thicker. So it's another level of complication, I will say. So a few years ago, we were very limited for cryotomography because we were only working in the very thin areas, the edges of the cells, like they are thin enough to be able to go to a transmission electron microscope without the need of cryo-FIPSEN. So we were missing all that area. This is crazy. Can you imagine all the information we were missing because we were unable to visualize that because it was too thick? And this is so frustrating. So finally, we had the idea of why not to do lamellas? Why not to mill away with the focus ion beam all the material like we don't care about, because we know where the area of interest is, because we have the light microscopy data and we can correlate and we can do the lamella in those areas and go to the transmission electron microscope and visualize all the thick area like before we cannot. So this is an examples like you can see, for example, here on the SEM, this is a lamella around probably 200 nanometers, and this is the visualization and then the TEM. So already there, you can already visualize plenty of details. So thanks to the FIP, people adopting tomography is increasing, As you can see here in the last probably 5 years, the growth, if you combine tomography with cryo-FIP, so the preparation of the lamella is almost 36%. And we are expecting more and more growth, okay? Plenty of people, like they are experts in cryo-EM single particle analysis, they want to go right now toward tomography because again, they want to visualize the protein of their interest in the context of the cell. And they can do that right now with the cryofib, and it's very cool. People doing tomography is increasing as well. When I say tomography without the cryofib, it's, for example, with bacteria only or with viruses, okay? Like samples, like they are thinner, like less than 300 nanometers. So you can still, you need to do tomography still with viruses or bacteria, the whole virus, the whole bacteria, but because they are thinner than 300 nanometers, you can go directly to the transmission electron microscope without the need of the cryofib. So that scientists are growing as well. You know, people like they are virologists, they don't need the cryofib, but they need the tomography area of that. So how you prepare, you know, the cells, and this is very cool. So this is the kind of results that you will be able to obtain once all the segmentation, tomogram, subtomogram averaging is done. So it's very cool. The first step, as I say, you grow the cells in your cryogrid. And this is the tricky part. Once your cells are very happy there, you use the VitroBot. Again, the same way I explained before with the single particle analysis, is the same machine to you know, vitrify those grids or vitrify those cells. So this is your grid with your buffer and your cells very happy there. You do the, you know, the vitrification process. So you are removing all the excess of liquid. So you have a very thin layer covering the cells. And after that, you are plunging very, very quickly and very fast into the cup with the liquid ethane, okay? And that is minus 180 degrees Celsius. While the cells are vitrified, you need to store them the same way like with proteins under liquid nitrogen conditions. After that, you need to go to the cryofib sen. And this is the typical instrument with the cryofib sen. So how it works to prepare the lamella. Oh, I went too fast. Oh, let me play this video here because this is very cool. So you introduce, this is the internal anatomy of the cryofib sen instrument. So everything is under cryo conditions. So you can introduce in this subtle 2 different grids with your cells, like they are vitrified. And after you have, you know, this is a spotter coating, like it's, you know, for protection of your sample. After that, you will focus in one specific grid with your cells. You will localize the cell of interest and you will be able to do the milling. So that's the electron beam, the scanning electron beam for imaging. And in red, you have the ion beam for doing the milling. These are gallium, okay? It's a gallium ion beam. So this is your cell in your grid. You can, with the software, automatically identify, you know, those yellow squares. Those are the areas that you want to mill away. And in between, you have the thin lamella in green, okay, around 250 nanometers. So again, with the focus ion beam, you will mill all the material away. You will create the lamella. And that lamella, you know, has the area of interest because, and I will stop here the video, because in this machine as well, we have an integrated fluorescence light microscope. So that's super cool because before we even had the integrated fluorescence light microscope. So that what means? So you have your Leica system. Usually people use the Leica system and they are very good. So you look at the thing, okay, you see, okay, the area I want is there. You transfer the shuttle with the cryo-EM grids into your FIPSEN. You prepare the lamella, but you are doing that blindly. You don't even know where is the area of interest anymore, and you are like praying like you are doing the lamella in the correct area. Right now, we have an integrated fluorescence light microscope that can help you with that because you can get back and forth to localize, okay, I am milling the area, I am still in the good area of interest where my protein is. If you can see the fluorescence, good to go. And you can mill in, and you can mill in, and you can mill in. And you can be back and forth to verify and double check, triple check, you know, if you want, like you have the area of interest in that lamella. So you are not doing the milling blind anymore. So that's super convenient. So once you have your lamella ready, you go to a transmission electron microscope with your lamella and you start collecting data the way I explained before. All the till series to create, you know, the tomograms. And after, when you have the tomograms collected, you will be able to do with softwares, tomogram averaging, subtomogram averaging, and being able to create, you know, all the segmentation work and visualize this, the ER, the Golgi, the ribosomes, the nucleus is super exciting. And even your protein of interest, of course. So let's show you some examples of people already doing this and very successfully. So of course, we are still in the middle of the pandemic. I think I will call it the endemic already because I think it's gonna stay with us as the flu, probably. So this beautiful guy here, it was, for 3 years, he's pissing us out. So yes, so we were able, using tomography, again, because it's a virus, we don't need to do the cryofib part, but to analyze, the spike. And this is very cool because you can see, how the spike is interacting, the composition of the spike, and etc. So this was a very, very well done work published in Cell in 2020. What happened with a cell is infected with the SARS-CoV-2 virus. And this is the thing that you can visualize again, because there are cells, you need to prepare the lamella with the cryophypsen. And once you have that, here you can see in the left, you can see already the tomograms, and you can see plenty of details here. You can see, for example, the virion here, a body virion coming here, another virion, another body virion. And you can, again, do the segmentation of this and generate the 3D reconstruction of all those parts in the cell, internally in the cell. So you can see the virions going out of the cell. So it's very cool. HIV, when I was a postdoc at Scripps in La Jolla, I was working on HIV for years, and other nasty viruses like Ebola and flu, all the very good guys. And this is the typical tomogram of the HIV virus. And you can see, because the viruses in general, they are very random. That's the reason you cannot do single particle analysis in the whole virus. You need to do tomography, okay? Some viruses, in particular for HIV, they have some viruses plenty of spikes, some other viruses almost nothing. And even in the same virus, you can see areas with a high concentration of spikes versus other areas with almost nothing, okay? So it's very random. So using cryotomo, we were able to obtain this beautiful 3D reconstruction of the virus. And it was the first time, quite a high resolution already for Tomo. I think it's around 4.55 angstroms. You can already see the first druggable pocket fully resolved in the whole virus. So again, this is very interesting and very cool because you can try to develop new vaccines and new strategies to fight this virus. More examples, this is the nuclear pore. And again, all this work was done by tomography. The nuclear pore, as you can imagine, is a very complex machinery. You have plenty of proteins in there. So good luck if you try to crystallize that, or good luck if you try to do everything by single particle analysis. It's not going to happen, or it's going to be a very difficult process. Using tomography, you will be able to visualize directly on the context of the cell these nuclear pores. So it's very, very cool. More examples here. This is again mammalian primary cilia, was revealed by cryo-electron tomography. It was the first time that you can see, the EP1 proteins in yellow, they are along the microtubules. So first time that this was visualized using cryotomo. More examples here. These are ribosomes. So it's very easy, or relatively easy, to have high resolution of the ribosome by single particle analysis. But what happened with ribosomes in situ, in the cell? So we are able to do this analysis, isolating all those little ribosomes here from the tomograms. And it was very cool. And you can see different resolutions there. I love this video. I don't know if some one of you are working with, sarcomer. This is very exciting because we are able to literally zoom in the muscle cells. And you have the skeletal in the right side, in the left side, sorry, and the cardiac in the right side. And let me play this video for you guys. So we prepare the cryofib milling of the cells. We collect the data, after the lamellas in the transmission electron microscope, and we can zoom in those filaments, and we can get a 6.6 angstroms of the skeletal one and 7.7 of the cardiac one. But if you really zoom in the core of the protein, in the core of the filament, you can really see a 4.5 angstroms, and you can already start understanding how this nebulin in pink is binding to the actin. And you can already see even some residues, we know they are playing an important role in this binding. all this is done in situ. So believe me, when I started doing tomography probably 10 years ago, the best you can get by cryotomo, it was probably 15 to 17 angstroms. And that was already like, wow, champagne, celebration. Right now, we are getting to 4 angstroms quite easily. You know, again, depending on your sample, okay? No every sample is the same. It's heavily sample dependent, but the technique is getting there. Ribosomes, again, I want to show you, like we were able to obtain, you know, 4 angstrom resolutions of the ribosome in situ. So this is your cells in your grid, you know, vitrify it. This is the way you prepare the lamella. You can see the lamella here. Those areas of interest where you can collect the data in the transmission electron microscope. This is the tomogram. You know, all those still series put together. So you have a movie. And after you do all the tomograms of tomogram averaging to get this you know, complete final data, and you can see already the nuclear pore, the mitochondria, and the nuclear envelope. And you can select all those little ribosomes, you know, and do all the segmentation work to get the 3D volume at the end for angstroms. So it's very cool. More work here. This is bacterial cell. Because it's only 300 nanometers, we don't need to do the FIPSEN again. You can, you know, purify the bacteria, vitrify that in the grid, and go to the transmission electron microscope and collect data. So this is a typical tomogram you will be able to see here. And when you do all the segmentation work and tomograms of tomogram averaging, you will be able to obtain the 3D reconstruction of the bacterial cell, you know, with the bacterial envelope and all the different organelles there. So again, super, super impressive. So this is a typical lamella. You will be able to get here around 200 nanometers in thickness. This is chlamydomonas. So you can already see different organelles here. And those black spots are ice contamination. It's almost impossible to have a lamella without any you know, crystalline ice. So you will always see these areas of black spots with ice. But once you put that in the microscope and do all the tomograms of tomogram averaging, etc., you will be able to get these beautiful things here. And, you know, all the ER, the Golgi, the nucleus, the ribosomes, and separate those last by colors. So all that segmentation work can be done with a mirror, and it's very cool. Again, it's still a bit manual, but it's very rewarding when you have that at the end. So, of course, this work was done not only internally in Thermo, but as well with plenty of collaborators around the world. So obviously I want to thank them. And very quickly, because I have probably a bit more than 5 minutes left, and I don't think you are very interested in this, but I think it's always nice to have that in your mind just in case. So microED, okay, where microED goes in all this mess. Okay, so I explained to you, you know, single particle analysis, like it's usually very high resolutions in between 1.2, 1.5 to probably 4 angstroms. And the size of the proteins is probably around 50 kilodalton minimum to maximum, maybe 1 megadalton, around those lines. Cryotomo, you can work with biggest, you know, biological samples, but the resolutions are a bit more limited still right now, you know, probably it's around 4 to 10 angstroms under cryo conditions. Microelectron diffraction is still a bit overlapping with single particle and crystallography, and a bit of NMR as well. So it's nice because you can get very high resolutions using very small crystals like before. You can put them in the trash because, you know, for crystallography, that won't work. So how it works, and this is the typical pipeline for microED. So the first thing you need to do is like have the crystal. So again, disadvantage of microED, you still need to have a crystal. Yeah, lucky you. So, but the positive thing is like the majority of the people doing microED, they are using small molecules or small compounds, natural compounds, like usually crystallize relatively quickly, okay? So that's good news. So once you have your crystal, okay, very small crystals, we are talking in between 200 and 500 nanometers. So they are very, very, very small crystals. You vitrify those crystals in the cryo-EM grid using the vitrobot, the same way you do with the cells, the same way you do with a single particle, okay? You go to the transmission electron microscope, low dose screening, you localize your crystal, and you see you have diffraction, okay? Because if you don't have diffraction, it's not a good crystal. If you have diffraction spots, you can already start doing a tilt series, data collection. So continuous tilt of your crystal to collect under diffraction mode all the information you need. So usually one crystal takes in between 3 to 5 minutes. So it's very quickly. And usually you probably need on average 10 crystals of the same compound to be able to get all the information you need to have, the 3D model reconstruction at the end. You use the same programs like crystallographers are using for decades for phasing and refinement. They are the same softwares. So, how micro-EV differentiates from the other techniques, So first of all, crystallography, typical crystallography, you need very big crystals, usually 5 microns, but 5 microns, and you need probably around 20 crystals on average, to be able to get nice resolutions. You can as well use XFEL. I don't know if someone is familiar with that technique. It's very cool because you can use small crystals, but you need to use tons of those crystals. In this case, for example, we need to have more than 80K crystals, very small crystals, to be able to get like 2 angstrom resolution. Micro-EV, the beauty is like you can use small crystals. You need a small amount of crystals, as I said, on average, 10 crystals of the same compound, and you can do that very quickly. You don't need to go to the synchrotron. You can do that in-house because you can do that in the transmission electron microscope. So some examples of, samples like the tau peptide, lysozymes, TGF, have already been resolved using this technique, microelectron diffraction. So again, if you have at some point working with crystals and they are not big enough to go for crystallography, don't put them away, save them. You never know, maybe it can be good for micro-EV. So another example I want to show you here, and this is, I think, probably my last slide, is like for granulovirus. This is granulin. It's a very small protein, 29 kilodalton. It's impossible to do by single particle analysis because it's too small. But you can see you can do crystallography or XFEL or micro-EV. So again, the difference is, again, the resolution, the best is always with crystallography, but you need to have, a relatively important number of crystals, like they are quite big, 5 microns by 5 microns. And it's not always the case. Sometimes it's difficult to get those crystals. The XFEL, you can use small crystals, as I said before, but you need a huge amount of those crystals. So micro-EV is a good compromise where you can still use small crystals, a very small amount, probably 5 crystals is enough, on average 5 to 10 usually. Okay, the resolution is still the worst of all the 3, but I think it's a good balance. You can use your microscope in-house without the need to go to the synchrotron, and you can still use those crystals. Okay, so I think think about it. And with that, I'm done. So thank you so much for supporting me for an hour.
Thank you, Natalia. We have time for a few questions.
I actually have 2 for you, Natalia. First one, is it a destructive technique or can you re-image over and over and over again?
Single particle,
Yeah,
Thermo.
single particle.
Yeah, single particle, usually no, you have radiation damage in your sample. So usually you take only one image in that specific hole, and that's it. You cannot reuse that grid over and over again.
Okay.
Yeah.
Second question is, all proteins, what about nucleic acids? Can you do all the same type of approach with nucleic acids?
Yes. We are working a lot right now as well with DNA, RNA, and not only DNA or RNA associated with proteins, but by itself. And that's very challenging because, as you know, they can be quite flexible. They can be elongated. So, it's always more difficult to do the 2D alignments with all those elongate samples, but we are doing that right now. So yeah, it's possible. Yes. Thank you. Yeah. Yeah, so right now with all the hardware like we have, so that's a very good question, because at the beginning you need to collect millions of particles to be able to have a 3D reconstruction like is accurate and with high resolution. Right now, because we have cameras, like they are very good, and softwares, like they have artificial intelligence, you know, and different algorithms, like can help you to really only collect in the very good areas of the grid. So you don't need to have millions of particles right now to be able to get very high resolutions of your protein, because of the hardware, because of the cameras we have, like we call the direct electron detector cameras, like increase dramatically the signal to noise ratio, you know, because in electron microscopy is always a balance in between contrast and resolution. So you need to be able to see the particles, but without losing the high resolution information, you know. So with all the hardwares and software developments like we have currently, and again, applying algorithms, machine learning, artificial intelligence, we need right now to collect probably 100K particles should be enough. And again, depending on your sample, if you have samples like are flexible or they have different conformations, so dimers, trimers, etc., it's going to be more challenging. But in general, you can reduce dramatically the number of particles that you need to collect right now. Yeah. Yeah. Yeah, we have, that's a very good question too. So we have plenty of algorithms, you know, when we are correlating the data from the light microscope into a transmission electron microscope, or the thing that we call the CLEN, you know, or even when you prepare the lamella, you need to have that correlation. So we have plenty of algorithms like can help us, you know, with fiducials as well to understand where is the area of interest and where we need to prepare the lamella. Or if you don't want to prepare the lamella and you are going directly to the transmission electron microscope and you are doing CLEN directly, we had that correlation because we had the algorithms and we had the fiducials. So yeah, of course.
Hi.
Yeah.
Very nice presentation. I really enjoy it. And,
thank you.
And the powerful technique. I wish I have it here. I wondering about proteins that are not very stable, like very disordered proteins. Mm-hmm. So you show us proteins that have a stable structure, but what happened with proteins that have disordered regions, and you can have that the same resolution?
Yeah, so the resolutions I saw when layout is the thing like we call the overall resolution, but usually when you are publishing a figure in a paper, you know, in cryo-EM, you usually have the local resolutions. So for example, the resolution can go in the core of the protein, like it's very stable, probably 3 angstroms, depending on the sample. And maybe some areas like they are flexible or disordered, you can go to 8 angstroms, you know, so the overall resolution can be 5, but you have local resolutions. Okay. Regarding your question, when you are having disordered areas, like you cannot really align, like you cannot really get an accurate 3D reconstruction in that area because it's very flexible or because it's very disordered, the thing that we are doing right now is integrating that with mass spectrometry. So plenty of people are doing native mass spectrometry, cross-link mass spectrometry to understand, you know, what is going on with those areas, like they are disordered or they are like, you know, very flexible. Right. So that's something like I will highly recommend if you have access to mass spectrometry core to try to combine both. And this is the thing that we call the integrative structural biology, like it's the toolbox of the structural biologists like me. Never use only one technique, use as many as you can. And mass spectrometry can be super helpful for that kind of proteins.
Another question is, you said that for small proteins it is difficult. So how do you think it I mean, there is some possibility to get that kind of resolution for small proteins, like a single molecule analysis.
Totally. So, for example, I have some customers like they are working with very small proteins, like for example, 70 kilodalton, 60 kilodalton, or even less than that. So the first thing I recommended is like trying to find nanobodies or antibodies like Fabs. You know, like they can bind specific epitopes. First of all, if the protein is too tiny, adding a Fab can increase the size by 50 kilodaltons, so can be already a huge improvement, and can help as well with the orientation of the protein, because you know where that specific fab, which is the epitope of that specific fab. So that can be super cool. Another thing, another trick that you can use is like if you know other companion protein, like this protein can bind to try to put those proteins together, so to increase even the mass. So nanobodies, antibodies, and trying to find a protein companion.
Right. Thank you.
We need to increase the mass. So
yeah, of course.
Yeah,
Yeah, thanks.
of course.
Okay, so just one last question. Okay, so can ask no.
So how can you discriminate like your protein of interest? You're looking for some small protein inside the cell. How can you identify and discriminate from other? I think this is a question more or less related to Philip's one and Rocco's. Can you bind some antibody
Yes.
with some metal?
Yeah. So there are different methods. Probably you are already familiar with immunostaining. So that's mostly for room temperature samples, but you can apply this as well for cryo. So one thing is like find a specific antibody, like it's only binding to the protein of interest very specifically. After you find another secondary antibody, like bind only to the first antibody, and that secondary antibody can have nanogold particles, for example. So that you can even visualize without the need of the fluorescence light microscope, because the gold particles under the transmission electron microscope, they are very black. And usually if you can work with different sizes, 10 nanometers or 5 nanometers, so you can really visualize very well where those nanoparticles are, and you know your protein of interest is in the area there. Other way of doing is having a GFP, for example, a fluorophore, something like binds to the protein of interest, and you can go to the light microscope and visualize where the area of the fluorophore is. And once you have that area, you know the protein of interest in there, you can prepare the lamella there and start data collection later.
So we can say that in the case of Thermo, in the case of small proteins, the application would be like localization?
Yes.
Okay.
Yes. Otherwise, it's going to be like, trying to find something very small in a huge house. It's going to be impossible. Yes.
Okay, thank you so much, Natalia.
Of course. Thank you.
So now it's time for a dancing folkloric dance. Folkloric dance, sorry. So the name of the group is Chopin, which means spring in Nahuatl, which is a native Mexican language. Chopin, at this time, is led by Claudia Zapata Nieto, and they will dance different special dances from Campeche, Jalisco, Chihuahua, Huasteca region, Guerrero, Sinaloa, and Nayarit. So please welcome Chopin group. Okay, so you have 5 minutes if you want to go to start your deck, and then come back.